think Kuvam and Julia, you two maybe will go first. I have the great joy and pleasure of introducing two people in my group who have been at ASDRP for over a year, and in some cases multiple years. I think, let's see, Kuvam, do you want to say who you are, and then Julia? And then we'll jump right in.
Hello, I'm Kuvam. I'm a senior at Washington High School, and I've been in Edwards Group for, I think, 1.5 years now. I'm going to the UC San Diego in the fall.
And Julia? Go ahead.
Okay. Hello, my name is Julia. I've been in Ed's group for, I think, a year now? A year? Yeah, a year. And I'm a senior at Aquarium, and I will be attending Stanford next year. Yeah.
Cool. So this is a very special evening for us as a group because we get to see, for one very last presentation, two just superstar people. I'll give a very brief introduction, then you two may jump right in. I think there's a running saying in this group that if there is a reaction that's worth troubleshooting, probably Kuvam can troubleshoot it. Just one of the best experimentalists I've ever mentored at ASDRP, bar none. One of the hardest working young men in this group who has helped spearhead a lot of really great science, including that which is now in a preclinical collaboration with the biotech company. So we'll be missing you, Kuvam, next year for all the crazy cool reactions that you do between 1 milligram and 10 grams. Julia has worked on a number of projects that are all very difficult projects, and yet she's never fazed by the fact that these are very challenging and tricky, and in some cases 95% troubleshooting, 5% success projects. But Julia will be speaking a little bit tonight about some of her work in medchem and hit to lead optimization in this group. So without further ado, go ahead and share your slides, and we're very excited to hear what you have to share with us today.
Okay, so I'm just gonna get started. Hello, we just introduced ourselves, but I'm Julia, and Kuvam and I will be presenting on our work in Edwards Group, particularly addressing different perspectives that we take on translational chemical synthesis to synthesize to target biomedical problems. And yes, okay, next slide. If medicine were like software, then organic chemistry is the boot code. This is allegedly a quote that Ed says a lot, although I have personally never heard those words escape his mouth, but it is something that is very true in my opinion. Organic synthesis is a field that is at the very heart of chemistry, and is a really key part of a wide range of industries with different branches of knowledge and ultimately changing lives for better. And in particular, the medical field, particularly drug discovery and development, is a sector where the impact of chemical synthesis is incredibly important to human life. And generally, as many of you might know, our group focuses primarily on synthetic organic chemistry. And generally, the main objective of synthesis is to look at is to kind of test different chemical theories, to look at, to try and develop a better or more efficient chemical compound, and determining the structure of materials when we extract it from a natural resource. And for this reason, chemical synthesis and its techniques are incredibly foundational and important in clinical and pharmaceutical studies. And it allows us to create, to look at different methods, and to look at, I guess the right way to say it is that chemists have the power to discover and optimize the synthesis of really complex compounds for its specific biological properties, and to use these innovative pathways to synthesize novel compounds that are usable for medicinal treatments, to suppress symptoms, to cure diseases. And it is something that we do a lot in our group. And so this slide here shows some of the many different things that we do in the group. Reaction methodology, physical organic chemistry, natural product synthesis, and chemical biology. In our group, we pursue various different research initiatives in which we incorporate computational modeling, like pharmaceutical chemistry, natural product synthesis, all to work towards developing molecular solutions to biomedical problems. And in our presentation, which is quite lengthy, I'm just going to put that in there. We are going to present on 4 of our projects that particularly explores sort of the interface of chemical synthesis and medicine, and highlights the different modern approaches to drug discovery and drug development. In particular, I'm going to be focusing mostly on optimizing certain properties of chemicals to create more effective analogs, whereas Kevin will talk more about sort of bridging the gap between the gap of like drug delivery and the importance of total synthesis to medicine. Slide. Okay, so this is a project that I am very, very involved in. And it is the benchtop 19F NMR spectroscopy enabled kinetic and mechanistic studies of the synthesis of carmofur and its related 5-fluorouracil analogs. And these are my collaborators, yes. So basically, just to give a quick overview, the molecule that you see in the middle right there, carmofur, is a prodrug derivative of 5-fluorouracil that was originally developed as an antineoplastic agent used in the treatment of colorectal cancer through ceramidase-induced apoptosis. However, more recent drug repurposing efforts published in Nature, I believe, reveals that carmofur is a novel and really promising lead antiviral compound in the inhibition of the SARS-CoV-2 main protease, Mpro, through covalently binding to the S-gamma atom of the catalytic cyst 145 dyad. And it is what our entire project kind of revolves around. Slide. So as I said before, the SARS-CoV-2 main protease is called Mpro, and it plays a really important role in really important role in the in protein production within the virus, and so its inhibition is the goal is to inhibit the main protease Mpro, and a really effective compound, as I mentioned earlier, is carmofur, as discovered through more recent drug repurposing efforts. And so essentially, carmofur covalently binds to the S-gamma atom of the catalytic cyst 145 dyad, and basically the catalytic dyad attacks the sulfur in the catalytic dyad. It attacks the carbonyl carbon of carmofur. I don't have access over the, yes, thank you. And this basically cleaves off the 5-fluorouracil head and the lipophilic aliphatic tail, as seen over here. It inserts into the S2 subsites and engages in hydrophobic interactions with the amino hydrogen side chains of which stabilizes the Mpro active sites, and the antineoplastic 5-fluorouracil is then cleaved off and functions to inhibit RNA processing and other viral functions of SARS-CoV-2, making it a really effective treatment for COVID-19. Slide. And so first we used computational homology modeling. And we basically used it to screen analogs of carmofur and to determine its binding affinity to Mpro and its mutated variants. And slide. we basically designed a library of 33 different analogs of 5-fluorouracil with varying different substituents to yield 3 different classes of analogs, those with amino acid side chains, those with alkyl side chains, and those with benzene side chains. And we kind of plotted them. Oh, slide. And first, basically, we docked them. And so what we did is that we looked at common mutations in the active sites of Mpro, and we looked at how our different analogs would, I guess, how they would interact with the active sites. And we looked at the different bonds that it forms with the surrounding amino acids, and what it would indicate as to how well the compound will bind to the active site. Slide. And so we did that for all of the 33, the library of 33 different analogs that we designed, and we kind of plotted them into this high thorough virtual screen, or HTVS heat map, such that the colors that are approaching red indicates a more negative binding affinity, and therefore a greater binding affinity to Mpro, making it a more promising antiviral agent for us to synthesize and look into. Slide. And as for docking results, we saw a general trend of when we increased the aliphatic tail length, it kind of correlated to marginally better binding affinity with Mpro, and for the majority of the variants that we studied, likely due to the increased number of hydrophobic interactions that the longer chain is able to form with the hydrophobic residues. Tryptophan, which is an aromatic amino acid, consistently had the greatest binding affinity amongst a variety of the wild type Mpro variants, and we hypothesized that it's because these amino acid side chains are designed to mimic the native peptide to yield higher binding affinity. And the benzene and aromatic analogs exhibit significantly fewer hydrophobic interactions. Slide. And so using this knowledge, we started moving more into synthesizing these analogs that we had designed and tested computationally. So we synthesized carmofur by coupling 5-fluorouracil with N-hexyl isocyanate in purity conditions, as seen in the schematic up there. Essentially what happens is that the nucleophilic NH in the 5th position of 5-fluorouracil attacks the electrophilic carbon in the isocyanate, and we use the same mechanism to synthesize a library of chosen carmofur analogs in good yields, and we hypothesize in particular that this analog on the top right-hand corner, right here, which was synthesized with chloroethyl isocyanate, will be an especially effective neoplastic chemotherapeutic as a result of the presence of the chlorine, which mimics the structure of nitrogen mustards, which have widely been used as an anti-cancer drug. Slide. So 5-fluorouracil, and so that raises a big question of how we're going to monitor this reaction. 5-fluorouracil is a very polar compound, and coupling it with an isocyanate makes it an even more polar compound. And due to the extreme polarity of carmofur and all the analogs that we synthesize, its synthesis can't be quantitatively tracked through more conventional methods, such as thin layer chromatography, because the polarity prevents its movement in TLC. However, its fluorine head serves as a spectroscopic handle for benchtop 19F nuclear magnetic resonance spectroscopy, or NMR, which we use to kinetically monitor the reaction of 5-fluorouracil with the various isocyanates. And more recent advances in benchtop NMR has facilitated the real-time monitoring, characterization, and quantification of these chemical entities, making it a really practical tool for precise structural elucidation and optimizing reaction conditions, and just tracking impurities and yields, as well as reaction kinetics, which is what we use it for. And furthermore, its protein lock capacities make the synthesis of carmofur not require the use of dehydrated solvents, which would be significantly less financially feasible for scalable synthesis. And we use 19F-NMR to enable the optimization of the reaction, which was seemingly impossible to track through quantitative means by conventional means to synthesize our products at gram scale. Slide. And so over here, you see our concentration versus time graph for all the different analogs that we looked at. And for most for analogs, we observed maximum conversion within 150 minutes with only 1.5 equivalences of isocyanates. However, in particular for our aryl isocyanate uracil coupling reactions, particularly as depicted on the screen, 3,5-dimethylphenol and Mpro, it did not react with that amount and overall produced significantly lower yields, which led to a series of studies where we looked at the differences between the reactivity of the alkyl versus the aryl isocyanates. And we hypothesized that reactions with aryl isocyanates have a lot more difficulty coupling with high-fluorouracil due to the conjugated pi bonds from the carbon the isocyanates that the 5-fluorouracil attacks in its aromatic ring. And to resolve this issue, we added additional equivalences of aryl isocyanates, and as seen in the triplicates below, with 10 equivalences of isocyanate, we were able to reach maximum conversion within about 120 minutes. And additionally, as seen over here, we actually discovered that water plays a role in the reaction. When we noticed that there was a significant amount of urea forming in our isocyanate bottle from the hydrolysis of our isocyanates, which led to a series of mechanistic studies, which you will see more of in the next slide. And so currently we're kind of looking at trying to probe the mechanism of the reaction. And so after observing the effect of the isocyanate quantity on the reaction progression, we initially hypothesized that the urea that was forming due to the hydrolysis of isocyanate acted as a catalyst for the acetylation. However, our experiments demonstrated that the water, in fact, did not improve the rate of the reaction, as shown in the graph over here. Talk about moisture and its effect on the concentration. We also looked at optimizing the reaction, and we have since shifted our focus more towards optimizing the conditions on which the synthesis of carmofur occurs, and just looking at its conversion under various polar aprotic solvents and under different temperatures. And so, as seen in the two graphs over here, all of these experiments were done in triplicates, and in the solvent experiments, in our solvent screen, we noticed that there is a significantly consistently higher yields in purity. And over in the temperatures screen, we can notice that at 90 degrees Celsius, there is initially a very high conversion, but very quick degradation of the reaction, whereas higher temperatures generally correlate to better conversion, with 45 degrees showing the best conversion at 150 minutes. We have also used fluorinated isocyanates. These are more current studies, but we're using fluorinated isocyanates to kind of track the catalytic effects of the hydrolysis in various other reaction conditions. However, we recently noticed that there was an intermediate that was being formed, which is something that we're currently looking into. And we believe that it might be the result of the formation of due to the trimerization and the dimerization of our isocyanates. And it is something that we're still looking into. Slide. And so in the future, what we hope to move forward is testing carmofur and all of its analogs biologically, and looking at how well it inhibits Mpro, as well as how well it does in colorectal cancer. And we plan to do this through either fluorogenic activity assays or colorimetric assays. Slide. And sort of moving on to another project that I'm very involved in, the synthesis of novel rivastigmine analogs as therapeutic leads for various neurodegenerative diseases. Slide. And these are my coworkers. So the enzyme acetylcholinesterase, or basically it inhibits the synaptic transmission by hydrolyzing the neurotransmitter acetylcholine. And the enzyme inactivation induced by various inhibitors lead to acetylcholine accumulation and the hyperstimulation of the receptors, which disrupts the neurotransmission. Generally, increased activity of acetylcholinesterase excessively breaks down acetylcholine, which results in various neurological conditions like Alzheimer's or Parkinson's degree, where the signals that are usually sent through the nerves are not do not occur. And so more recently, scientists have looked into inhibitors of acetylcholinesterase to inhibit the enzyme to prevent it from breaking down acetylcholine and increase both the level and duration of the neurotransmitter action. Slide. And one of the major modern challenges that scientists and medical professionals face while looking into the chemical synthesis while looking into chemical neuroscience is the ability of the medicine to penetrate the blood-brain barrier, which is the barrier in between the brain's blood vessels and all the other components that make up the brain tissue. And basically the blood-brain barrier, it protects the brain against circulating toxins or pathogens that could cause brain infections, while all the same allowing nutrients to reach the brain. However, the vast majority of potential drug treatments do not readily cross the barrier, which is a huge, as you can imagine, a huge impediment in treating mental and neurological disorders. However, physostigmine, which I will talk about in a little bit, is a cholinesterase inhibitor that crosses the blood-brain barrier and reversibly inhibits cholinesterases in both the central and peripheral nervous system, which thereby allows acetylcholine to accumulate within the synapse. Slide. For centuries, neuroactive alkaloids isolated from naturally occurring sources in nature have been very crucial in the identification and optimization of the synthesis of small molecules with the possibility and the potential to treat neurological disorders. And some of these compounds have gone on to clinical uses themselves, but the vast majority of them have inspired the development of but the vast majority of them have gone on to kind of inspire the development and the synthesis of synthetic analogs, which might possess greater potency or better pharmacological features than the natural product itself. And one such alkaloid is physostigmine, which is found in the alkyl bean plant. I believe Physostigma venom, honestly, I'm not even going to try, but which is found in the calabar bean plants shown right here. And it is demonstrated to be a really potent cholinesterase inhibitor. Slide. In particular, physostigmine, it possesses N-methyl carbamate moiety, which reversibly acetylates the active sites serine in acetylcholinesterase, which renders the enzyme inactive and allows for the and prevents the breakdown of acetylcholine and other acetylcholine and butyrcholine. And as a result, this causes acetylcholine accumulation near the synapses in the central nervous system, which triggers the action potential and enhances the effect of the acetylcholine. And so as seen here, we can see the similarities between the structural similarities of acetylcholine and physostigmine. And so essentially, physostigmine has the ability to, as the active site see here, you can see here, it has the ability to acetylate this active site and prevents and act as a competitive inhibitor for acetylcholine. Slide. However, despite its potency as an enzyme inhibitor, physostigmine has a very physostigmine, its low bioavailability and its short half-term life and its acute toxicity has really limited its therapeutic potential, which prompted the development of rivastigmine, as you see here, which is essentially a synthetic aminocarbamate small molecule that is inspired by and structurally very similar to physostigmine. And in, I believe, 2000, rivastigmine was approved by the FDA to treat neurodegenerative diseases such as dementia, Alzheimer's, Parkinson's. And acetylcholinesterase, which is the enzyme that breaks down acetylcholine. Slide. And essentially, acetylcholinesterase, which is the enzyme that breaks down acetylcholine, has these two binding sites for acetylcholine: the anionic binding site and the esteric binding site. And rivastigmine acts as a competitive inhibitor similar to physostigmine, and it binds to the enzyme's active site in a very similar manner to acetylcholine, which prevents the hydrolysis of acetylcholine by binding to the esteric part of the catalytic site. And the acetylcholinesterase then cleaves the rivastigmine molecule and releases the resulting phenol. Slide. And so the research in our group is focused on the synthetic optimization of rivastigmine and its various analogs, utilizing computer modeling and biological assays to determine the most favorable analog for the inhibition of acetylcholinesterase and get more structural insights into the biochemical behavior of these compounds. Slide. Okay, and so first, we worked primarily on optimizing the synthesis of racemic rivastigmine, or basically to be racemic, it means that it's a mixture of both the S and the R enantiomer of rivastigmine. So previously reported synthesis of rivastigmine involved the acetylation of 3,1-dimethyl aminoethyl phenyl and N-ethyl N-methyl aminocarbamyl chloride in the excess of sodium hydride with relatively high yields. However, sodium hydride is a very pyrophoric compound, which makes it really difficult to handle due to its high sensitivity in air and moisture. And so we aim to identify alternative conditions to synthesize rivastigmine and its various analogs that would avoid the use of sodium hydride, which led to our efforts in optimizing the synthesis of rivastigmine. And so our efforts originally began with the acetylation of which was accomplished with pyridine and DMAP in DCM, which resulted in about 57.3% yield. And we also tried synthesizing rivastigmine without a base, using a base pyridine, but its effort was generally unsuccessful and showed no conversion as observed in TLC. And so we basically just looked at its reaction in different solvents and using different bases as catalysts. Slide. So after we worked on the optimization and the synthesis of rivastigmine, we started looking into the optimization and the synthesis of its three analogs. And so essentially, we see here how we used the library of different analogs that we synthesized using N-N-dialkyl carbomoyl chlorides. Slide. As well as the synthesis of SN analogs of rivastigmine, as shown here. And basically, I'm not going to go too much into depth because we don't have much time, but we basically use that same mechanism to synthesize and optimize the synthesis of a wide variety of analogs. Slide. Something we also worked in is computational models. And computational studies allow us to kind of predict the behavior of our carbamate compounds while testing their efficacy with in vivo and ex vivo assays. And docking studies were used to kind of determine the binding affinity to these compounds. And from these results, we discovered that physostigmine had the lowest binding affinity, followed by, if I'm not mistaken, S-morpholine, and its R enantiomer. And the molecules that performed really well had various interactions with the acetylcholinease binding pockets. Physostigmine demonstrated both aromatic and both hydrogen bonding interactions with the best performing conformer, notably, as shown up here, notably aromatic face-to-edge pi stacking, a parallel displaced pi stacking, as well as various hydrogen bonding. And we also looked at the binding interactions of the R enantiomer and the S enantiomer of all of our analogs, which also showed very similar aromatic and hydrogen bonding. And we use this to determine the binding affinity, similar as we did with carmofur and its analogs. Slide. And so Lipinski's rule of 5 is, or more simply known as just the rule of 5, RO5, is a rule of thumb that is used to kind of evaluate the drug likeliness of a compound and to determine if a chemical compound with a certain biological activity has chemical properties or physical properties that would make it an orally active drug in humans. And so the rule was originally formulated by Christoph A. Lipinski in, I believe, 1997, and it is primarily based off observation of the most orally administered drugs are relatively small and moderately lipophilic molecules. And the rule kind of describes, I'm not going to read it out loud, but the rule kind of describes the molecular properties that are important for a drug's pharmacokinetic properties inside a human body, including its absorption and its distribution, metabolism, and excretion. And these rules are really important to keep in mind while looking at drug discovery and while looking at a pharmacologically active lead structure. And so we calculated the log p value of each of our compounds, which is the logarithm of the partition coefficient between the N-octanol and water log. And this is a well-established measure of the compound's hydrophobicity. And low hydrophobicities and therefore high log p values, which would cause poor absorption on permeation. And we did that for all of our carbamate analogs. And additionally, we also use topological polar surface area, or TPSA, which is defined by the structure of the surface of the polar atoms in a molecule, which we use to analyze drug transport. And compounds with high TPSA are transported well, while those with low TPSA are not. Slide. Additionally, we also performed extensive colorimetric assays to test the biological activity of our compounds, and in particular, our compounds physostigmine against acetylcholinesterase, and such that, as seen right here, the more yellow the solution, the more effective inhibition. And physostigmine was shown to be the most effective inhibitor of all of our novel analogs. Our compound dimethyl. Our dimethyl analog exhibited the highest level of inhibition, Our rivastigmine diethyl and morpholine analogs were observed to have minimal effect on the inhibition of acetylcholinesterase. As a result, were not significantly different from the control observance value. And through such studies, we determined that rivastigmine analogs are significantly less effective in inhibiting acetylcholinesterase than physostigmine. However, this discovery prompted us to pursue two different routes, both the synthesis of S-enantiomer versions of our analogs with the goal of making more potent analogs, as well as the study of biological activities of all of our analogs on both cholinesterase enzymes to determine enzyme selectivity. Slide. Since physostigmine was found to be the most effective inhibitor, a synthesis of a diethyl analog of rivastigmine is likely to demonstrate higher inhibitory activity in the rivastigmine itself against acetylcholinesterase. Additionally, rivastigmine is a dual cholinesterase inhibitor with no selectivity between acetylcholinesterase and butyrylcholinesterase, and it is possible that our analogs might exhibit more selectivity towards one over the other. And additionally, it's been reported in the past that S enantiomers of rivastigmine is more biologically active than the R enantiomer of rivastigmine. And so our current synthesized analogs are a racemic mixture, which is both of them combined. And so whether the role of the stereochemistry can be seen in our analogs still remains to be seen, and it is something that we hope to work towards in the future. Slide.
All right, so Julia has talked mostly about a lot of small molecule therapeutics, and in the world of therapeutics, there's like two main classes of like therapeutics. One of them is small molecules. One of them are therapeutics. The small molecule is basically like anything which is like small, like rivastigmine or like aspirin or like a lot of the other compounds which are typically available over the counter and stuff. And then there's also the biologics, which are like more of the type of molecules that are made in an organism, such as antibodies, antibody drug conjugates, peptides, or like mRNA vaccine, like the COVID vaccine. And specifically, I'll be talking more about the antibody drug conjugates in my part of the presentation. So basically, what is an ADC? So currently in a lot of modern cancer therapies like chemotherapy, there's this issue where the cancer drugs are not really very selective, and so you go through chemotherapy, you lose your hair, you lose your immune system, stuff like that. And so after your immune system gets weak, you can easily like get sepsis from any stray disease, and that causes a lot of deaths too. And what ADCs basically does is that it tries to solve this issue of drug selectivity by having an antibody which goes towards an exact antigen, so it'll bind to only one antigen. And so because of that, it solves the selectivity issues as the antibody works as like a delivery vehicle for the drug. And so basically an ADC is just made of an antibody, some kind of linker which combines the antibody and the drug, and the drug, of course. And so here is one ADC which our group is currently working on. And this ADC specifically is an enzyme releasable ADC of MMAE. So MMAE is this very cytotoxic molecule, which basically is like, it cannot be administered by itself because it's just that deadly. And so we're using an ADC to increase its selectivity, and so it would only target the tumor site. And so the way that this thing works is that we have a linker, the antibody, and the payload MMAE. And so what happens is that after it enters the cell, some kind of enzyme will cleave it at this amide over here, and that would trigger this whole mechanism that releases the payload. And once MMAE is released, then that would trigger apoptosis inside the cancer cells and kill them off. And some important things to consider over here is here's also the synthetic route for which we make it inside. And yeah, it's just a pretty simple synthetic route using a lot of relatively cheap molecules, and so it is pretty scalable. And that's part of the question which we need to consider when actually making these ADCs, is that one, like, what is the design of it, how scalable is it, how safe is it, and like what is that kind of the yields, because whatever is made is with the goal of sending it to clinical trials one day, and so it has to be scalable and made inside large enough quantities to be able to one day support like some kind of industrial use of it. And so that's one consideration for these antibodies. Another one of these ADC, and another condition to think about is like, how are we planning on releasing the drug. So this one, for example, uses an enzyme releasable method, and another one which I want to talk about is the photocaged ADCs. So that is one of the things which we specialize in this group and have spent a lot of time working on. So what this basically does is that you have the antibody, some kind of linker, and the drug like usual. And what's special about photolabel ADC is that you have this photolabel protecting group over here on the linker. And so what happens over here is that you have this O-nitrobenzyl group over here, and when you shine UV light at that, then that would release this whole cytotoxic payload. Oh, my bad. So for example, over here you have a cytotoxic drug called protophyllotoxin, which again can't be administered by itself. And so by having an ADC of it, we increase the selectivity of this molecule so that it only target the tumor sites. And I guess something special to note is that in the past there have not been any photocaged ADCs released. So this is just like a list of all of or many of the ADCs that are FDA approved currently, and yeah, none of them are photoreleasable. So, and yeah, there's also one example over here of this thing called Mylotarg, which also is not a photoreleasable ADC. This is a different type of linker. And so how these photolabel ADCs work is that you have this photolabel protecting group, or like which makes like an O-nitrobenzyl ester, or in our case carbamates. And so basically the way this works is that you shine UV light at this photolabel protecting group, and then it goes to the starred mechanism and it releases the payload. And yeah, we have worked on some optimization work for this thing because what we wanted to do is that we wanted to make the fastest possible rate of photo release and control the rate of photo release inside our molecules. Because if the rate of photo release is like a lot faster, then what we can basically do is that we can shine light at it for less time and release like more drug, and that makes it a lot more efficient in using this method of drug delivery. And so we tested many different functional substitutions on these aromatic rings, like the methoxys, dimethoxys, and the bromos, so including withdrawing and donating things. And through these computational studies, we've figured that like having donating substituents on the 4 and the 5 position, we can increase the rate of photo release. And so like the 4,5-dimethoxy over here predicted to do pretty well for that. And this is just a list of the intermediates that it goes through when doing photo release. So here's the beginning of this excited state, and then it goes to this at the very end, releasing the payload and creating this other thing from the photolysable group. And we've also have some future plans on optimizing the rate of photo release a lot more through this thing called the kinetic isotope effect. And so this basically says that we can replace a hydrogen on a photolysable group with a deuterium, and then what this would do is that if that step where the hydrogen gets taken off, or the proton gets taken off, is the rate determining step, then that would affect the rate of photo release. So like if we have the deuterium there and then let's say it gets slower or faster, then we know that that's like the rate determining step, and that would also determine if we can have a benzoic substitution over there to affect the rate of photo release. And so that's something which you can look into like later on. So we have tested these, or applied these photolabel protecting groups to drugs before. So for example, we have protophyllotoxin over here, which is, as I explained earlier, a cytotoxic payload. And so what we did over here is we made like a prodrug of it. It's not like an ADC just yet. This is just like a prodrug, so it's simply like the photolabel protecting group and the drug. It's not like connected to antibodies yet. But this does have a similar purpose of increasing drug selectivity, because this way we can shine UV light at any part of like the body, like for example this like the hand or whatever, and release the drug only over there, so you only release whatever is like in circulation over there and at the tumor site. And yeah, so this basically like was like a proof of concept in making our prodrugs, and for later on using the same technology for ADCs. And yeah, we also did some docking studies over here just to like see the binding affinities of these prodrugs, because one extra part about making a prodrug is that not only like is it supposed to be like releasable somehow, but also when you make the prodrug of the molecule, then the molecule should not be able to be as potent anymore, but otherwise there's like no point in making the prodrug, because it's just still gonna do the same amount of harm to the body. And so yeah, we did these docking studies, and it was kind of surprising to us to figure out that by computationally, these prodrugs of protophyllotoxin were actually like better at binding to the binding site than protophyllotoxin itself. So I guess like we either made a prodrug over here, or it's like just a better analog. It's a win-win either way. And Yeah, so after that, we did take this whole thing to making an ADC, and yeah, this was actually a very long effort, along with a lot of troubleshooting and different synthetic routes and stuff. So this is like one of our very first synthetic routes that we made like about a year ago, and basically the main idea of this is that you have some kind of spacer in between the drug, which is like this blue thing. It can be any drug, and that would like connect to the photoacceptable group, and that would connect to the antibody. And yeah, the main idea was that we just have like a long linker, and that would I guess we can attach that to an antibody and use that. But yeah, this method ended up not working when we actually applied it. So the way that this would normally work when we use this ADC is that you would have this photoacceptable group over here, and when you shine UV light at it, then this carbamide would cleave over here, leaving an open amine, which would do an O-NaCl shift, so the amine would attack this ester over here and release the drug. And you just thought that was like a pretty cool idea of releasing the drug. And yeah, when we tried that out, we had this synthetic route over here where we basically plan on making this ADC, but we ran into this issue where when you like deprotect one of these protecting groups over here on the nitrogen, then like we ended up having the O-NaCl shift happen too early, and so that way we just couldn't make the molecule. So we had to like come up with like a completely different synthetic route over here, and so we thought, okay, what if we just make this whole linker but in like a different way? So over here we have, so instead of like having a aminopentanoic acid linker like over here, we have a double glycine linker, which basically works the exact same way. You have an O-NaCl shift over here where you release this carbamide over here, and that would attack this ester over here and just release the payload. And yeah, again, one of the issues with this entire route is that we figured that we were spending too many steps with the drug, so that would like that was just not like scalable at all, and also like you'd cause and also like just we had acylation issues with our drug, because like we had this synthetic route over here, we used diphosphine or like triphosphine or something like that to like create our carbamates, and that just ended up like creating a lot of side products, which was just really unfavorable for us. On top of that, like for this drug over here, we have like this was quinidine, I believe, and we reacted that to block glycine, and later on we reacted that to something else. That was two step with the drug, and let's say we went to like a more like expensive drug like MMAE, then that would just not be scalable, because like even if you get like 70% yield on two steps on each of two steps, like that would be 70% of the 75% that you have at the very end, and so this is like a very low yield. It's not like scalable at all. So we use a different synthetic route, and this is the final route which we like came up with and we're currently like using. So the way this works is that we have our photoacceptable group over here, and we propagulate it in our first step, which sets it up for a late stage click reaction, which is like another highlight of the synthetic route. And after that, we create our carbamate using nitrophenylchloroformate, and then attaching our cytotoxic payload, which basically creates like a prodrug over here, and it's also just like a linker which we can attach on to an ADC. And then after that, after we make this part, we just set aside that linker, and we take this azidopectidine malamide. And so the malamide is basically the group which attaches on to the antibody, and the way that does this is that this alkene over here would do a Michael addition with thiols on open cysteines inside the antibody, and that would just cause the linker to attach to the antibody. And so after that, we can just do a click reaction and make our ADC. And so far, yeah, we've reached compound 5, and we're currently working on and troubleshooting some methods of attaching it to an antibody. And currently, one of the main issues with this is that we are trying to be able to characterize our ADC, because we can't just use typical methods of characterization like NMR. We have to use mass spectrometry, and that just requires method development, stuff like that, and we're currently working on the troubleshooting for that. And this is also a future plan for this ADC thing, where we're using a near IR releasable linker. So what's different about this linker compared to the previous one is that here you just had like a nitrobenzyl group over here as our photoacceptable group, and here now you have like a O-nitrobenzyl styrol protecting group. So like it's like a nitrobenzyl group over here with some pi conjugation and some a donating aromatic system. And so what we've figured out from previous literature is that by having such a donated system, then we would redshift the photo release and cause the photos to happen more near IR like red light. And so the advantage of this thing is that UV light, it can't really penetrate the skin very far, but IR can. Like IR can go like a lot farther through the skin than just UV, and so because of that, we can like target more types of cancer and go like farther inside of the body when targeting different types of cancers. And yeah, so along with this thing, I guess one of the main ideas of organic chemistry that have came along with this is just a lot of reaction troubleshooting and stuff like that, because it's not like we just make something, or like we want to make something and then it just happens. It requires like a lot of troubleshooting and creating new synthetic routes and just a lot of troubleshooting in general. And so that's also especially related to this other multi-step synthesis which I had called and this is on the synthesis of cyclophilin E, which is an anti-proliferative molecule found in a deep sea fungus called the Aspergillus versicolor ZLN60. And the main reason why we're making this compound is because it's very inefficient to isolate this molecule from nature. It takes about 100 liters of this fungus just to create like 30 milligrams of the compound, and so we don't want to like just go to the deep sea and like just kill an entire like species of fungus just to get like a small amount of this molecule to be able to test with. And so instead, what we wanted to do is that we wanted to make this molecule from more simpler and cheaper molecules like amino acids. So like it would just antinucleic acid, L-proline, and N-acetyltryptophan. And so what we could do is that we can just combine these three molecules and make cyclophilin E. And yeah, this molecule has been synthesized in the past through this paper reported on the JOC. And I guess one of the things over here that was that we wanted to change is the use of a lot of like dangerous reagents and stuff like that. So first, and so like they use palladium over carbon, or like isotopic anhydride, or like potassium fluoride, which is like potassium fluoride is pretty dangerous, and same with palladium over carbon. Hydrogen gas is just really flammable and stuff. And so like for scalability, we thought, okay, what if we use much safer molecules in the synthesis of cyclophilin E? And we also did want to make some more molecules in the future, more analogs of the cyclophilin E in the future, such as this 3-amino benzoic acid analog. With like an expanded cyclophilin ring, and that could not have been done in the past, especially because of this isotopic anhydride, which they used as their antinucleic acid derivative, which is basically like a cyclized version of antinucleic acid, and you cannot have a cyclized 3-amino benzoic acid. And so by using our new route, we wanted to make a safer route and also have more possibilities for analogs in the future. And so this is basically a simplified route which we initially came up with, which basically uses a derivative of antinucleic acid, methylene to begin with, and then you Bock protect that, and then you react that to N-acetyltryptophan, and then you just react with the proline afterwards and make cyclophilin E. And I guess the key step over here is that we have this indole N-acylation step over here with the antinucleic acid and the indole NH of the tryptophan over here, which is basically just a very difficult acylation to do. Like we have a hindered electrophile and a hindered nucleophile also, which there's just very little literature reported on syntheses of indole and benzoates, and so we spent a lot of time troubleshooting this reaction over here because it's just very, very difficult to do. So like here was just our initial acylation attempts for making these indole and benzoates. So we just decided to use like common coupling agents like EDC, HA2, HBTU, PIBOP, and TBTU. And yeah, this ended up just not working. I guess it makes sense because they're just very sterically hindered molecules. And so yeah, from there we decided, okay, let's use some test substrates that are less precious, and let's just do some methodology study with this. So we have a molecule similar to the tryptophan, this indole 3-acetic acid methyl ester, and then we have a fluorine tagged benzoic acid derivative over here, which is kind of similar to the antinucleic acid that we have. And so from this we could track a bunch of reactions from 19F NMR and figure out which conditions work and which don't, and stuff like that. But yeah, we ended up having this issue where just nothing worked, and like any attempts to isolate these molecules, these hypothesized product molecules, just did not work at all, and like it produced such negligible amounts of compound that we just could not even use it. And so from here we decided, okay, let's maybe revisit some of the past synthetic route compounds, such as isotopic anhydride. And yeah, we had some trials with this, and we tried replacing the potassium fluoride with other bases just to like, make it simpler and less dangerous, such as potassium carbonate and all, but like that just did not work. And so yeah, we did get to get the reaction to work with isotopic anhydride, except like we did find this issue where the heating for this reaction had to be very consistent. Like you have to have it on like 60 degrees for like 21 hours straight. Otherwise, if it was slightly uneven, then the whole reaction was decomposed. And so we found this as a very inconvenient thing, especially for scale up and all. And so we did want to like revisit our other method of acylating the tryptophan. And so we had this idea of using acyl halides, which is like a lot more reactive than carboxylic acids, like on this bulk antinyl acid over here. So we thought, okay, what if we make this into an acyl halide? Like, what if you use like pentafluoropyridine and turn this into an acyl chloride, or like use oxalic chloride and turn this into an acyl chloride? And yeah, for this thing, we actually ran into this issue again, where like the reaction condition would not work. Where so when we reacted our bocantinic acid to pentafluoropyridine, we just had this issue where after the first intermediate, we just were not able to like form the acyl chloride. It was just that sterically hindered. And in oxalic chloride, we noticed that this actually forms a lot of other side products, and so we just could not use that method either. And so we realized that when we create our acyl halide, then we just have this thing where we can either cyclize our Bach ring, or we can just remove the Bach itself, and that kind of ruined our whole synthetic route. And yeah, we also figured that, okay, if the Bach cyclizes with these acyl halides, what if it cyclizes with other conditions too, like our previous coupling agents? And yeah, it actually turned out that that was the case, where when you use like a lot of these coupling agents like HA2 or HBTU, we just cyclize our indole, our Bach antinyl acid, and that was the reason why it was not working. And yeah, something to note was that when we use EDC, which is a weaker coupling agent, we just created an EDC activated antinyl acid, and that did not cyclize. And then from there we decided, okay, if changing the reagents don't work, let's maybe change the mechanism around. So here we decided to use some very strong bases and stuff, like sodium terbutoxide to maybe deproton our indole, and that would make it a stronger nucleophile. And from that we were hoping that, okay, maybe now we can get our acylation to work. But yeah, again, this did not seem to work at all. And yeah, we decided, okay, what if cyclization is not the issue? And yeah, that deprotonated indole could open the ring, but yeah, again, that did not work out properly. Like, just anything we tried did not really work out. We got some negligible amounts of compound on as like a hit M over Z on mass spec, but like any attempts to isolate whatever product we formed just produced negligible yields, and so we could not really proceed with those routes. And so yeah, we decided, okay, let's troubleshoot even further. We can, if changing the mechanism does not work, changing the coupling agents does not work, let's try just changing the reagents around completely. So instead of having a Bach protecting antinylic acid, we had a nitro protecting antinylic acid. And so what we decided to do over here is that we just use a smaller protecting group, which would decrease the steric hindrance from this whatever amine protecting group we have, and then that way we can maybe acylate this endoamine a lot more easily, and then after that we can just deprotect that nitro with tin chloride and create and just proceed with our route normally. So yeah, we had this huge table of conditions to make this compound, and I think some of you might notice that we use this much stronger base than previously. We normally use triethylamine and stuff like that, but yeah, again, we did try the standard acylation conditions; those did not work, and so we tried these more extravagant conditions, like stronger base like DBU, or stronger nucleophilic catalysts like Hobbit or Hoat. And we also did find some previous literature things that kind of corroborated with possibly using these conditions. But yeah, it seems like this one actually showed some kind of conversion happening, but yeah, it turned out that the conversion we had was actually just decomposition. And so yeah, under typical acylation conditions that we were using in the past, the tryptophan has just been decomposing, and so I guess after all this troubleshooting, we basically just validated that the past synthetic route for using isoic anhydride is basically the best way of making this indole and benzoic compound. And so if you end up making cyclophilin E in the future, we'll be using that route. And yeah, we did have some future plans for this if we do make cyclophilin E, which includes a lot more analogs, possibly a glycine analog or a sulfonamide analog of cyclophilin E. So for the glycine analog, we've changed this proline ring over here of the proline and just replace that with the glycine, which the main goal for that is that we would make the molecule a lot more flexible, and that would allow for more conformers of the molecule. And so currently we have this thing where you just do not know what the molecule binds to, but we have this hypothesis where if you have a more flexible molecule, we can maybe make more conformers, and possibly one of those conformers can have a much higher binding affinity to whatever cyclophilin E binds to, and so we wanted to make this as a possible future analog. And we did some computational modeling to just show that increase in conformers too. And so these are just some DFT structures of cyclophilin E and the glycine analog with their lowest conformer, lowest energy conformers. And yeah, we did have an idea, possible modification of the antinylic acid by making a sulfonamide analog. This is also just something which we were considering, and we were thinking that maybe we can try this in the future too. And yeah, back to our computational work with the glycine analog and cyclophilin E itself, we did also do some RMSE values stacking of the two analogs, and just to like see if the lowest energy conformers were like similar enough. And so like if this and so we were basically just seeing how similar the lowest energy conformers were of cyclophilin E, and if the similar conformers had like lower energy with the glycine analog. And we also had like a future plan of possible ring expansion. So instead of using a antineoplastic acid, we have a 3-amino benzoic acid, and so we just make a larger ring of cyclophilin E, and that was just a possible idea for more analog cyclophilin. And yeah, here's just some like key takeaways for I guess organic chemistry in general. So I guess the one thing which is just needless to say is that organic chemistry is hard. It takes a lot of troubleshooting for everything. Like basically just any research plan, just a lot of troubleshooting, and then you finally get some small success. And yeah, there's a lot of failures within organic chemistry, which basically just makes it. It's just like the reality of science. There's a lot of failures, and you gotta like keep troubleshooting everything, and based on this troubleshooting, you eventually build success on your failures. And just from all those failures in the past, it just makes success like all the more rewarding. Like in organic chemistry, like any reactivity setup is like 9 out of 10 times it's not going to work on the first try, but like when you finally get something to work, or like finally get a whole synthetic route to work, it's just like all the more rewarding. Like I remember the first day that when we like managed to make our ADC route work, we were just like screaming with joy and like in lab, just like looking at our MR, like, oh, we finally got this to work. And yeah, I guess one of the things which just kept me going is that the project was just really cool. And like at the end of the day, like we did have some times when like we were just depressed and like, oh, nothing works, but like I just remember that, okay, this project is really cool with solving cancer, and that's why like I just kept going. And yeah, I guess some other takeaways is that just read literature. There's always stuff in the literature that can help you with troubleshooting, and also just, yeah, a little bit of rigor goes a long way. Just seeing what happened in the past, and maybe you can figure out, like for example, our cyclophilin stuff. We looked at some of our past NMRs, and like because we have like some rigorous protocol with this, and we like knew what we did for everything, we could figure out that, oh, something is decomposing, and we could figure out what is happening. And so yeah, just a little bit of rigor goes a long way. And yeah, that's basically all we have. I'd like to acknowledge ASDRP and the All Children Foundation and all of our other supporters. And I'd also like to thank Edward Njoo and the rest of our group. All right, thank you. Any question, guys?
I think Julie's already been tackling people in the chat window, but any questions? Maybe people can unmute if you have questions. And then we have a second presentation, which hopefully won't take this long. But
Should we start with the ones in the chat, or do we just?
well, sure. Or people can unmute and verbally ask questions if they so desire. Either way.
Hello, folks. Can you hear me?
Hey, Clinton, nice to see you here.
Hi. Yes, it is I. I just had one quick question about the antibodies, because I've always wanted to find like an economic way to kind of create antibodies. So maybe you can talk a little bit about that, because that's always been like a goal since my undergrad bio labs.
Are you saying like creating antibodies, or like antibody drug conjugates?
I guess like, how would do you have like a supply of like unlabeled antibodies that you add the drugs to, or do you like order custom ones, or like how does that work?
Yeah, so we have like a combination of both of those. So like to begin with, we have like some custom, like some antibodies like pertuzumab, which we order like IgG, that we just wanted to like test on. And yeah, this whole like project was going to be a collaboration with Sunny Bay Biotech, and so we're currently like in a collaboration with them. Like we're just and they're gonna like be giving us their antibodies. Will be like making ADCs of it and sending it over. And so like it's like we have a supply of antibodies from Sunny Bay, but we also have like our own like other antibodies to test on. So like currently we're in the phase of just like testing this whole technology, just making sure that our conjugation actually works. So we're using like other antibodies like pertuzumab and all, and after that we do plan on using Sunny Bay's antibody, which will basically be like our source of antibodies.
Nice. No, congrats. That's awesome. Thanks for the clarification.
Other questions? So Clinton, we also have a hybridoma cell line that mass produces antibodies. So we can harvest them at will at some point in time in the future once that's optimized. Yeah, it's pretty exciting.
Yeah, that sounds amazing.
Yeah. Okay, other questions? Other questions?
I see one in the chat saying, how does NMR work? So basically NMR just works by like, it kind of like, so like instead of any molecule, like, let me just go back a bit. Yeah, so like for example, cyclophilin, E, right? Like it has a lot of protons on it along with carbons. Like every single one of these, like I guess corners over here has like a carbon along with like hydrogens paired up to it. And so what NMR basically does is that it like shines some radiation at these hydrogens, and then what happens at the hydrogens just like start like moving around and and then it like releases some like stuff back. And so based on how these hydrogens like I guess vibrate, we can tell like what functional group is next to, like if it's next to an amide, or if it's like next instead of some aromatic ring, it vibrate differently and release different like I guess signals. And so from that we can just tell what molecule we've made.
Okay, in the interest of time, I think we should go on to the second presentation of the evening. So our second set of presenters here. So thank you, Kuvum and Julia. Those of you who have questions for Kuvum and Julia in particular, maybe they could leave their email in the chat window, and then we'll move on.
Yeah, thanks, Ed. So this is Suresh here. So I belong to the computer sciences group, and today the team will present a data science model for detecting diseases. So I will hand over the presentation to Shivi and her team. Okay, Shivi, go ahead.
All right, so our presentation won't be that long compared to the other one, but we just want to talk about the research we've been doing during this spring session. So Anj, can you share your screen?
Yeah, give me a second.
All right. All right. Hi, my name is Shivi Narang, and I'm a sophomore at Mission San Jose High School.
Hi, my name is Sriram Natarajan, and I'm also a sophomore at American High School.
Hi, my name is Anj Singhal. I'm a freshman at Archbishop Midi High School.
And during the spring session, our research project was preventing infant and maternal mortality by analyzing cardiotachograms. So what will we be covering today? So first we'll talk about the introduction, which will include the problem, our current methods, and the solution we present. Then we'll go over data collection, more specifically the sources we used, and the actual analysis of the data we conducted. Then we'll talk about the model, including the methodology and the model building. And finally, we'll cover next steps, our future plans, and acknowledgements. So let's start off with the introduction. So what is infant mortality? Infant mortality is defined as the death of an infant before their first birthday. And the infant mortality rate is the number of infant deaths per 1,000 live births. The infant mortality rate is an integral marker for the health of society and reflects on the maternal and infant mortality trends. Until the beginning of the 20th centuries, the chances of surviving the period of infancy would rarely exceed 80%. However, today, the rates vary in different countries. In developed countries such as Australia and France, there are roughly 5 deaths per 1,000 live births. However, in underdeveloped countries like Nigeria, the infant mortality death rate can face up to 76 deaths per 1,000 live births. Here's some more infant mortality statistics. So in 2019, the infant mortality rate in the United States was 5.6 deaths per 1,000 live births. And in 2018, almost 21,000 infants died in the United States. In addition, 3 million newborn infants die each year, most due to preventable or treatable causes. I want to reiterate that last part where it says most due to preventable or treatable causes, because essentially this is the problem that we're going to be discussing, and we're going to look at the causes now. So what are some causes of infant mortality? So first, there's birth defects, preterm birth and low birth weight, injuries such as suffocation, sudden infant death syndrome, and maternal pregnancy complications. And these are the 5 most common causes of infant mortality. So this is the solution that we currently have implemented for preventing maternal and infant mortality and assessing it early on. And it's cardiotocography, or CTGs for short. And CTGs are simple and cost-accessible option to assess fetal health. They allow healthcare professionals to take action in order to prevent child and maternal mortality. The equipment sends ultrasound pulses that can be interpreted to shed light on various data points. Some of these data points include fetal heart rate, fetal contractions, dilations, etc. And this data is necessary to visualize the unhealthiness of the embryo, and it gives an opportunity for early intervention prior to a permanent impairment. So in our research session, we focused on preventing infant and maternal mortality by building a machine learning model to analyze CTGs efficiently.
So secondly, we have the data collection portion of our project. This includes our data resources as well as our data analysis. So what was needed for our data collection? We needed data from cardiotocography exams conducted by healthcare professionals that are used to prevent child and maternal mortality. We used the 2,126 records from the cardiotocogram exams to classify each feature into the 3 fetal health classes. There are 22 columns of data that we have classified into the 3 fetal health states. So what are the 3 classes or fetal health states that we keep talking about? These are the normal state, the suspect state, and the pathological state. To classify our data into the 3 states, some of the features we tested were the fetal heart rate, fetal movement, and uterine contractions. A lot of the data that is tested by a cardiotocograph is measured in seconds, which is why some of our numbers in our data are very small. For example, there's approximately 0.01389 movements per second in a fetus, and the healthy uterine contractions are between 0.005 contractions per second and 0.00833 contractions per second. So our data source is Kaggle, which is a dataset library owned by Google. Some of the characteristics of this dataset are the data is divided into 3 classes, which is the normal, suspect, and pathological. The data consists of the cardiotocography data for each class. The data is from a study from the Journal of Maternal, Fetal, and Neonatal Medicine. And lastly, there are 2,126 measurements from infants that are less than a year old.
The model. So here are some common concepts and terms used in our model. What is NumPy? NumPy is an open source math library that provides an assortment of routines for operations on multi-dimensional array objects. NumPy arrays have a fixed size of creation, and you will need to delete the array to change the size. The elements of a NumPy array will all have the same memory. It helps with calculations on large numbers of data, and many Python-based packages use this library. Pandas. Pandas is an open source Python package that is most widely used for data science and data analysis for machine learning tasks. Some things that we can do with pandas are data cleansing, data normalization, mergers and joins, data visualization, and statistical analysis. XGBoost was used as the model for our predictions. It is an open source machine learning software that allows us to implement regression into our project. It works through gradient descent as well. It works through gradient boosting as well as decision trees. Decision trees are given data and split the data into smaller groups. These groups can be narrowed down to ask more specific questions to allow for more accurate results. The branches are assessed and decisions are made based off them and their responses. As shown, an image of a decision tree is found on the right. The decision tree is trying to predict an animal species. It asks a basic question if an animal has feathers with options of true or false. If it is true, it asks another true or false question, such as if it can fly. Eventually, decision tree will find a result after multiple true or false statements. Gradient boosting combines multiple models as predictions into one to make a more accurate prediction. It uses a gradient descent algorithm to reduce its loss function by finding the local minimum. So as you can see on the right and the bottom, there is an image of gradient descent. Gradient descent tries to find the minimum value on a parabolic-like system. The minimum value is the loss function, and in the case of the image on the right, it is the minimum costs. So this is an example of our model's code. We use fetal health as our dependent variable. We assign this to Y. We then split our test data into 33% of our data set. As shown on the right, we wanted to run the model and make our own predictions. To do this, we needed to input many different values into the model to make a prediction. We started off with a dictionary full of values for all the columns, and then generated random values into each column. We then fed it into our XGBoost model to make 50 different predictions as shown, zeros, twos, ones.
Next steps. So these are our future plans and a few acknowledgements. In the near future, we plan on submitting our research paper to a high school journal and work together to get our work published. We have already narrowed down some of the journals we plan to look into. Some of these are the Journal of Research High School, Journal of Student Research, and many more. We want to thank Mr. Subramanian for guiding us through this session and ASDRP for providing us a platform to collaborate together. Thank you. We're open to any questions.
I have one question. That was an excellent presentation, very succinct, and this is awesome work. How do you define the extent of false negatives in your set?
So, Shriram, do you have any false negatives?
Yeah, I mean, I believe that's something we have to look into more. Could you please elaborate on that?
Well, I think, so how are you defining what is a true positive and a true negative, I guess, when you're training your model?
Basically, our XGBoost models, I believe it uses complex math functions and like different types of functions to handle an input and take care of the false positives. I believe it's just built into the system for XGBoost. That's why it's widely and commonly used.
Got it. Thank you. Do you know what machine learning model XGBoost is using? Like some sort of neural network that is embedded into the system? Or
wait, can you repeat that, please? I didn't hear what you said.
What sort of machine learning model is being used in XGBoost? I'm not familiar with what XGBoost
I believe
is.
XGBoost is a type of classification system. And like I said earlier, it uses gradient boosting and different types of decision trees to function.
Just in case anybody is curious, this is basically what Ed asked. So how do you detect false negatives and so on, right? So false negative and false positives are terminology which is used in binary classification when you only have two classes. Okay, so that's the first thing. So here we have three classes, so there is no real definition for a false positive. Here what we do is we find the accuracy that the model can predict a particular class. That is how we define when you have more than two classes. And I don't know whether the team has created, but there is a thing called confusion matrix, which basically defines how confused the model is when it tries to predict a particular class. So team, like in case you want to go further and then build a confusion matrix, that will be useful.
Yeah, that is definitely something we plan on doing in the future.
Sure. Okay.
Any other questions?
There's a few questions in the chat window for you, so maybe you can answer those.
So I think we chose the XGBoost model specifically because we believed it would be like the most efficient, most accurate for our model. Yeah, so that was our main reasoning behind using XGBoost, but we could look into using other models if we wanted to get a higher accuracy. Yeah.
Yeah,
And,
we were, yeah, sorry, you could go.
okay, and regarding like the question about overfitting, there wasn't necessarily that much overfitting, like considering the fact that we had over like 2,000 sample like cases for data. And so we didn't really face that problem. To answer the question that just came in the chat, so far we have not tried any other models other than gradient descent and decision trees. I think during our previous project, we solely used decision trees. And so the gradient descent was something we were like trying this session, like trying something new. But I do think we're gonna look into like more like advanced models as well in the future. So the other question in the chat, what other factors would you look at if you would expand on this? One thing I think we focused on, especially in this data set, was that focus specifically on infant mortality. And like I defined earlier in our presentation, that's the death between like ages 0 to 1. However, we can expand our model for child mortality as well, which is from ages 1 to 5. And I think that would also like help out a lot more and bring more of an application to our project.
So to answer the question that just came in the chat, so in our code, we like in our model, we decided to like do like a practice prediction, and with that we are able to see like what the states are, or like what the classes are, like when we try to predict what classes all of the features would fall into, and from that we use that the result of that, and we use the result of that to evaluate our model. So we got like 0, 1s, and 2s, and the 0 represents a normal fetus, a 1 represents a suspect fetus, and a 2 represents a pathological fetus.
So the distribution of the data set was, I believe, we used 33% of it in our testing data. We split it up into 33%.
Yeah, so basically just for reference, normally when you're building a machine learning model, you have training data and testing data. So out of the data set, like Shriram said, 33% went into the testing data, and we use the other 66% for training. So I don't think we know the exact number of 0s, 1s, and 2s, but we do, it was a pretty even data set. So I think we do have it written down, but like, we don't know by the top of our heads. To answer the question that just came in the chat, we did have to do a lot of data analysis. For example, lots of our data, for example, like fetal movement, it was in seconds. And considering the fact that normally fetal movement is like measured within a period of like 10 minutes or more, those numbers were very like small, like in the hundreds of thousands. So we did have to like analyze those and see, like figure out the normal values for those as well.
So to answer one of the questions that just came in the chat, there were 22 columns in the data set. So in each of those 22 columns, there was like a specific feature that held data. So yeah.
And no, we did not compare our results against AutoML platforms.
We will look into that after this session. Any other questions?
Yeah, so to answer the question that just came in the chat, there are other features, as we mentioned, there's like the uterine contractions and the fetal movements that the CTGs are able to track. So using all of those, all of that data, we were able to make our model.
Yeah, and to add on some other factors that we like Anj said earlier, there were 22 different columns. So some other ones were like accelerations, decelerations, fetal movement, uterine contractions. I think there was also short-term variability within the actual like data itself. So yeah, those were also things we added to our model.